start. So today we'll be talking about AlphaFolds, which I think is like one of the most exciting things that happened in 2020, and probably the decade as well. There's so much high praise from biologists and chemists about this, and I think the machine learning community is also pretty hyped that they finally got to solve something that is like of such important real world impact. So let's get straight into it. I guess since we have experts on like biology and stuff here, I will just try to like just point out if I'm saying anything wrong. I am not an expert in either of the background or like the model itself, I think, even though I was trying to read a lot of papers and related materials, I feel like there's just like a rabbit hole that you can go into for AlphaFolds. It's really an incredible paper, I think, and you could spend so much time just reading it and related materials. So I guess to just motivate what we're doing here, there's some backgrounds, and then I'll also provide some like cool applications of it. First of all, what problem does AlphaFold solve? It's the problem of protein folding, which is to predict the structure of proteins from amino acid sequences. And so this problem, I guess, arises from like the central dogma of structural biology, which is DNA transcribes into RNA and translates into proteins. And then, in the middle here, there's like amino acids, which are encoded by the DNA. And what they look like is they have like sort of this big thing with hydrogens and nitrogens on top. And then there's a group, which is like this R group here, which defines what the amino acid is. And for example, it's like, it could be like a little chain, or like it could hold on to a ring. And when you have multiple amino acids together, then they form peptide bonds. And then magic happens when those little things that hang down from them starts interacting together. And then you have intermolecular forces that just spontaneously make this sort of beads of amino acids fold into a really complex 3D structure, which we call the protein. So, why do we care about proteins? Why do we care about protein folding? Turns out that basically every single living function in your body is produced by proteins. And then we also know that the structure of a protein is really, really important to inferring what the function of it is. So, for example, there's actually a postulate that you could replace some of the amino acids in the protein, and then as long as they sort of still form into the same shape, then the protein could still function like the same way as before. And that is why sort of a lot of genetic mutations end up being like just benign and not doing anything, because you could substitute amino acids. But then sometimes these things do have a bad effects on you, and you could have misfolded proteins that cause a lot of degenerative diseases like Alzheimer's, Parkinson's, Huntington's, etc. And that is why we care about predicting the structure of protein just from the amino acid sequence. So then here I have like a slide of just common proteins in case it's hard for you to like grasp it without concrete examples. So everybody knows what hemoglobins are. They're like what makes your blood red. they carry iron particles, and these are sort of the iron particles that are they're called hemes that wedge into the hemoglobin. And this is your first example of like how the form serves the function, where there's little sites where the heme groups can just insert into. I find proteins to be really cool because they're like little cogs machines that just are shaped so perfectly that you can like wedge other things into. For example, again, enzymes, they help you digest food. And basically how they do that is they take the substrates from the chemical reaction that breaks down foods like this, and they just like wedge into it. And that shape helps it to speed up the chemical reaction. And then polymerases is super cool because the protein folding and transcription and translation process is actually done by protein. So it's like there's like a self-referential quality there, which is very interesting. And then so polymerase is like this sort of like a zipper thing that hangs onto your DNA helix, and then it breaks it into two strands so that other things can operate on it and translate it. And then you have like antibodies that protect you against COVID. They're like Y-shapes. And again, their form serves their function as well here. So, and I guess another important thing, just one last thing to mention is there's this guy called Leventhal who is like a Nobel laureate, and he famously posited this paradox of how proteins fold, in that if you look at the search space of all possible foldings of an amino acid sequence, then that search space is actually bigger than the entire universe and the entire time that we have to sort of calculate through all of those possibilities. But then an actual protein just folds within milliseconds. It's like they know what to do. They somehow know how to fold like billions of different proteins. And it's all encoded within like such a small thing. So, and then Leventhal was like, okay, then maybe there's a way to directly tell what the structure of a protein is just by the DNA encoding or like the amino acid sequence, which is just going to be like, like a text string, like, so that becomes our problem, which is input a text string that is like, I don't know, like AZHP, and then output some kind of encoding of a 3D structure. And we'll see that AlphaFold does this output by sort of calculating the angles for each of the residues, which is what we call amino acids in the protein. But anyways, I'm getting ahead of myself. So as this is the exciting part. I talked about this, how good actually is AlphaFold. So there are existing technologies that tell you the structure of a protein. For example, you can shoot X-rays at it and then sort of crystallize the protein. That helps you identify angles easier. But then this method takes 3 to 5 years to perform just for every protein. And so people were, they started this computational competition, and it's called CASP. So then like for years since 2006, programs have been, algorithms that have been trying to crack this problem. And you can see that the accuracy isn't that great. So in comes AlphaFold onto the scene in 2018, right? And then they got about like 50, 58, I guess, something percent accuracy. And that was really good. And they won the competition, right? And people were very impressed. But then they came back and they were like, we're gonna put all of these biologists out of their jobs right now And 2020, they won the competition, but not only that, they reached like almost 90% accuracy, which is considered, like basically the problem is considered solved because now you can just go to AlphaFold's database and then look at a protein and it'll be like almost perfect prediction. So this was like absolutely groundbreaking. Yeah, and then here's like a totally memeable content of AlphaFolds too, like just reaching this insane level, while the rest are like trailing behind it. So this is their database, and I think it'll be a very fun exercise for us to like just go in it and I don't know, type your favorite protein in the chat, and then we will see what it looks like. And for now, I will just, while I'm waiting for the chat, I'll click a random thing. E. coli. Cryptokinase. Yeah, so this is the 3D model that is predicted by AlphaFold. I think it's just really nice to like look at these 3D structures. And this is the original amino acid sequence. And then they also tell you an expected error that is returned, sort of calculated as a loss function in the AlphaFold model. So these are all like very fun things to look at. I'm gonna check the chat. Sonic hedgehog protein. Is that a real thing? What's wonky protein? I don't actually know. Okay, I'm just gonna check more of the examples. Could catch, right? I don't even know how to pronounce that. I'll check it. Oh, it looks like they don't have it. Or maybe it was a different name. Could catch me. The only thing about this database is it's not very good at search results. Can search on here. Yeah, no. Sadly, we don't have it yet. It is an ongoing project, and they said they're looking at like the human genome database, and they're predicting every single protein that is produced by the human genome. So it's ongoing. One day we will have your sonic protein. Yeah, and then there's more fun stuff, which is they had a collab notebook. So I guess you guys can refer back to the slides for this link, but for now, because it has to like download a bunch of stuff, I will run it first. Oh, I guess I ran it beforehand. So this is our sequence here. And it's still running. Yeah. So this is the preliminary step where they're searching actual genetic databases for it. And then I guess AlphaFold is just running now. We will come back to it. So let's talk about applications of AlphaFold. So these are very domain specific, and forgive me if I don't know anything about them and butcher what they actually are, but basically I think what I got was there was a group of people who visualized the COVID-related proteins with AlphaFolds. This is their results, and So the green part is like the experimental results, whereas blue is what's predicted by AlphaFold. And then a major thing also I saw was accelerating drug discovery. There was something about druggable proteins, which I think are like proteins that are able to be like to have drugs take effect on them. But if you know more about this, feel free to speak up and elaborate. And then also, I think the most fun application that I found was there's a group of scientists who are like developing enzymes to break down plastic, just based on AlphaFold's predictions. They look like hair strands. Yes, they kind of do. Because essentially it's like a strand of amino acids. And then, so like you can see some what they call secondary structures, which are like these helices. And then also there's some like just sheets that go, you can see like parallel to each other. These are like the most common structures that you can see in protein. So then a quick search on like U of T's library helped me find a lot of papers in biology just discussing what they could do with AlphaFold. And for now, I haven't seen any like substantial results yet, but there's been a lot of discussion of like the promise of AlphaFold in chemical therapeutics, drug discovery, and then what we talked about, misfolded proteins. There's a whole survey on what AlphaFolds could do. And then my favorite is, can we AlphaFold our way out of the next pandemic? So apparently the visualization of the COVID protein this time was only sort of like an additional piece of information that didn't really help in the creation of the vaccine. But there are methods that use in structural biology where you see the structure and then directly you can infer a lot of properties about, I don't know, the related virus or the protein. And that could be like immensely helpful. Right, so then this is the model, the actual architecture of AlphaFold, specifically the version they used in 2020. And I'll have to admit that this is like a huge model, and the paper itself on Nature is 20 pages. And it's like you can tell that it's so condensed that they've like probably edited through 100 times just to fit the words count. And then every 2 sentences, they're like, please refer to supplemental methods. So I took a look at the supplemental materials and it's like 40 pages. It's a real rabbit hole, but I will give you like sort of the high level idea. And then later on, I'll open up the floor to discussion and then people can ask like about specific things, I guess. So like, first of all, there is what they call like sort of the pre-processing stage, where 2 important things are like databases are consulted. First of all, this is called MSA. It's short for multiple sequence alignment. And basically this is coming from an evolutionary point of view where some proteins are decoded from DNA. And DNA, we know, is genetic material that mutates throughout evolution. So what you can do is you can compare the DNA of related organisms through evolution. And then this becomes sort of like if you put all those sequences against each other in a row, and then you sort of insert gaps in between them, because mutations could take the form of like inserting something new or deleting something. And then the goal is to match up all of these sequences as much as possible by inserting gaps and like shifting it around. And this is actually an NP hard problem, which is very interesting in itself. I think for AlphaFold, they just like use the state-of-the-art solver for MSA. But anyways, so they have this evolutionary information. And then here, what is called like structured database search with templates is sort of going the other way around. It's like, let's take some protein that was already solved, which structure we already know, and then we compare it to its original amino acid sequence to the one that we're currently trying to solve. And then now we have this sort of like two information sources that we can reference. And then you see this like idea of having two information sources prevail through this entire model, in that this Evo former here, which is like an attention-based model. And if you don't know what attention is, you could refer to my November video, or I'll just tell you now. Basically, like there's like, you have Q, K, and V matrices, and then you have like a softmax function, which basically helps you index into a text sequence, or in this case, an amino acid sequence. And then it helps you pay attention to different parts of the text and associate like a kind of property, a value to those different parts of the text. So that, for example, like attention mechanisms first appeared in transformers for solving language models. So they were used in translating like text in natural language processing. And what you can imagine what you need in translation is like, you should pay attention to, for example, the grammar, where like complex phrases, multiple words that form the same word, or like you should pay attention to pronouns that refer to the same subject. And this is what attention does. It pays attention to different parts of your data. And so you have this like 48 blocks, repeating blocks of this transformer kind of, it's like a specially designed thing just for AlphaFold. And this transformer, which is shown in more detail here, this is like the block that we showed before. It takes into account both the MSA representation, as you can see in the top row, right? And as well as the pair representation that we got from the structural templates. And then there's some interactions here. For example, this pair representation arrow going into the row-wise attention, and then this outer product mean going backwards to the pair representation. So essentially you have like these two information sources influencing each other, and then sort of making the model better, like in this iterative process of like relating back to each other and solving the contradictions. And then to go a little more detail into this, so here we have like row-wise self-attention and column-wise self-attention. And all this is saying, let me try to find this slide, is, so here we have like multiple sequence alignment, like I said, like this picture at the bottom. Like I said before, you have like, you know, these sequences in rows, and then in columns, you can compare the mutations and differences and similarities against all of these. So row-wise attention will help you extract information about every single one of these similar evolutionary amino acid sequences, whereas column-wise tells you what mutations happened and sort of like how they changed throughout the MSA. And so this is how these are helpful. And then in the pair representation, this is a really interesting thing in AlphaFold, I think, and which raised a lot of doubts in the more traditional biologists and chemists, which is this triangle update. And the sort of justification behind that is it's all about the triangle inequality. So like if you have Euclidean distances, you better have like, so for example, you have point A to point B, and then going to point C, then this distance summed up better be no greater than this distance from A directly to C, right? And what they found was actually that AlphaFold was not able to deduce this very simple physics rule. And so they had to add like all of these blocks to say, oh, well, okay, now look at A, B, and C, and then, oh, maybe this A and C edge is even missing, so we have to add it, and then make sure that physics is preserved in our model. And that raised kind of a lot of doubts in people's minds, but I guess my take on it is just like this model doesn't think in the same way that you do, and what seems obvious to you is probably not obvious to it, and vice versa, right? So I guess that's something like curious about this model. And then so we see again like two flows of information going into what is the final block, the structure module. And what the structural module does is sort of beyond me. It's okay, so like I understand like the IPA is the invariant point attention, and what is novel about it, I guess, is that it is invariant to translations and rotations. So basically you have a function that calculates some value, and then no matter how the input is rotated or translated, it gives you the same value. And then this is helpful for this part of the structure because at the end, we want to predict angles between the residues in the protein. And so, yeah, if anyone can explain this part to me, I would be very glad to hear that. And then, so a final fun part is this recycling thing here. So this recycling came from the idea of self-distillation in machine learning, which is like a really wonky, wacky concept, which says, okay, you take a neural network, you take its predicted labels, and then you feed these predicted labels back to the neural network as if they were the ground truth labels, and train the network again, and then somehow it is able to produce higher accuracy on the test data than if it trained on the actual labeled training data. And then there's some missing details. For example, like you used to have to augment your predicted labels so that they're not exactly the same. But somehow this gives you a huge performance boost. And then there are proofs showing that this is because there is sort of an enhanced regularization effect when you do self-distillation. It sort of forces your neural network to condense its knowledge to be able to produce the same correct answers as before, and also generalize just a little bit because you've augmented the data. So I thought that was a very interesting concept to recycle what you had before. And I believe in the paper they said that the self-distillation method, it doesn't actually improve the accuracy that much, but it does sort of like iron out some inaccuracies in the physics or chemical constraints. So I think that's about it for like the high level idea. And if you were to like read the actual paper, there's some helpful terminologies here that I explained. And this is sort of what I talked about. Yeah, and then, oh, and this is just like an extra point about like their architecture in 2018 versus 2020. The main difference was in 2018 they used ResNets, and then they used gradient descent to iteratively improve upon their prediction. There's actually a very cool video showing how they went from this to this just during gradient descent. Oh, and this is like another interesting thing about publishing on a Nature scientific sort of journal instead of a pure machine learning journal. Is like they put so much emphasis on interpretability that you have to be able to explain what your neural network is doing. And that's sort of the fatal flaw of machine learning, that and also the benefit of it, that it's a black box and you just, it crunches numbers and you can sort of say like, oh, this is just the inner product of like vectors in space, whatever. But it's not really giving people insights on how the protein folds, how it comes together, and then what chemical forces are driving it together. And that's like sort of also a major objection to AlphaFold from the chemist's point of view. What was I saying? Yeah, right. So AlphaFold 1, they use resonance, basically. And then AlphaFold 2 used attention mechanisms. Even though attention is usually associated with like language processing, right? Somehow it was able to achieve a better result. And I thought that was very interesting. And actually to look at the interpretability parts of their supplemental like methods. Let me see. This one, 12 pages, right here. So they actually tried to show what each attention layer is trying to do. And what they found was that, for example, the layer 0, we see a pattern that is similar to a convolution. So it is doing convolution inside an attention, a transformer model. And somehow it's also doing other things that is helping it. Yeah, that is sort of, I think, all that I have to say about this. There are some like really great resources here that I really encourage you to go to. For example, they've released their open source implementation. You can play around with it. There is, they have, right, this is like the project to translate every single genome in the human genome database. And then the supplementary methods, oh God, it's hell of a read, it's 40 pages. But then they've got detailed equations, they've got algorithm details that would really clear up any sort of doubt you have about the details. And I think this just finished running. So let's take a look. Oh, I'm bad at zooming. Okay. Okay. Okay, I'm just bad at zooming. Give me a moment to figure it out. Oh, okay, that's yeah, that's acceptable. So this is our prediction for the amino acid sequence that we just entered. And here they've color-coded it in terms of their confidence, some predictions of errors. And yeah, it's just a really fun thing to run and see. And it also like downloads a zip file of a bunch of additional information. So play around with the collab and the database. And I would love to hear your thoughts on the paper now.
Oh, thank you for your presentation.
Thank you for
Mine
coming.
is not it's not really I don't have anything too much to share, to be honest. I didn't read this paper in too much detail. I read a few basically other blog posts or papers explaining this paper.
Mhm.
Um, so I think I guess like one thing that I can comment on is that the result is actually quite good. So the AlphaFold team has said that like it's not accurate for basically all the protein, right? But then specifically they gave the SARS, the COVID protein ORF8
Yeah,
as an example, saying that, okay, hey, this one is really challenging. And at the time, people thought, okay, like I think at the time they had one structure uh, being being assayed in the lab, and then that structure basically was a little different from what AlphaFold has predicted. But then they did it again. It turns out that AlphaFold actually provided a lot of uh, uh, like AlphaFold's prediction was actually a lot similar to the second prediction. At least that's what I remembered. And then it was, I think, as another example, like some researcher in Berkeley was like, okay, your prediction is a little different from mine. What's going on? But then they went back to the lab result and then figure out that, hey, like there's actually something wrong with the lab experiment, and then the AlphaFold was correct.
wow.
Um,
Yeah,
so
same.
yeah, so it's actually, I think it's kind of crazy
It is.
that we are able to do that.
Mhm.
Um, yeah, and then it's like they also did it for like a new completely new set of proteins too, which is pretty impressive.
Yeah, yeah. The ability to generalize is insane. Um, but also,
There is, yeah, sorry.
keep going.
Right. So I think there is this paper that came like many years ago when I was doing research, saying that we actually have enough data in our protein database uh, that basically covers many possible protein arrangements. I think it's not mentioned in the AlphaFold paper, but the basic conclusion is that um, our existing knowledge uh, covers a lot of configuration, and then it's just a matter of using them wisely. And then, so I thought that was kind of interesting that because and honestly, as a biologist or let's say as a chemist, we would think that protein has so many configurations to form,
Mhm.
uh, and actually the actual process of protein folding is much more complicated than just like okay spitting out a 3D structure. Sure, that's one thing, but it has to do with your environment. It has to do with the chaperone protein, which is another protein that helps you fold, and
Mhm.
then and the other like let's say for um, let's say in our actual cell where protein folding is not like hey like all of a sudden we have a structure that's laying out there and then start folding itself. It doesn't happen like that. The translation the translation machinery, which is the process of making protein, would gradually spit out one amino acid after another. So you can sort of imagine that in the physics world, protein folding starts to fold, I think around I forgot it was N terminal or C terminal, but it's not like the way that we're thinking computationally.
Right.
So like I actually don't know what's going on that made this so accurate, but this is very fascinating.
Yeah, it's like it's got a completely different way of thinking.
Yeah, and then I'm not sure if anyone understand the triangle multiplicative update part. Um,
Um,
like that's the part that kind of confuses me.
this part, right? I think
Yeah,
the big idea is to just sort of make a configuration that is physically possible, um, like with the triangular inequality. But like the details, I haven't gone into them either.
Oh, I see. Okay, so you're basically saying the pairwise. Oh, I see what it is. So the input. So okay, so the input matrix is not necessarily a pairwise. Basically, the pairwise distance doesn't really satisfy the whatever.
Yeah, it may not. It doesn't have an application to. And yeah, and that's a really weird thing.
But like, why would that be the first case? What would that be in the first place, though?
I think because the representation, right? The pair representation, it's just a matrix telling you every distance between every pair, but there's no constraints on it saying that, oh, if these two are close together, then it also affects a third.
So, so we are saying that AlphaFold would process the matrix in a way that it would generate basically an unrealistic structure, but then we have to like do some update to force it, back to like the physics world.
Kind of like that, yeah.
Why would that happen?
So I think the reason is like, if you look back at this diagram, right, your initial configuration for the pair representation comes from the MSA representation, and they influence each other in the way that like what you get from the MSA might not be a physically realistic picture, but then what you get from a pair representation might not actually match the evolutionary reasons. So that's why you have to sort of like go back and forth correcting for those.
Oh, so you're generating, oh, I see, okay. So you're generating the pair representation along the way.
Yeah, it changes along the way. Wait, going back to like what you said about all proteins can sort of just like be a few structural templates put together. Is that actually true? Because if that's true, then why aren't more like other computational methods more successful? If they're just like, try this template and then this and just enumerate over all of them.
Let me find the actual paper. I think I have that saved somewhere.
Yeah.
I might not have worded it correctly.
Oh, I keep forgetting about this, but since this meeting is recorded and this discussion is actually very helpful, would you be like willing to appear in the recording?
Don't know how much how helpful I am, but sure.
Thank you. You're very helpful. Does anyone else have any ideas on this paper?
Oh, I think, okay. So let me actually send, okay, I can send this link to you and then maybe we can look at it together. So
Yep.
I think it's pretty interesting that to kind of mention this. Yeah, I sent it to the chat. So the idea is that proteins have like basically proteins have like more than one domain. Like one domain is one functional domain that does one thing, like let's say bind to the DNA, like catalyze on the reaction or whatever.
Mhm.
But obviously like a a protein can contain, let's say, more than one domain. So that's why the sequence is pretty long. Like hemoglobin, I think it has 8 or something. But then let's say if we restrict our proteins to a single domain protein, then the sequence will be a lot shorter. And then there is a chance that our database would contain all possible configurations, all possible combinations of amino acids for a single domain protein. Maybe it's not as complicated as a multi-domain protein.
Mm-hmm.
So I think like there's a lot of terminologies here, but I think this one is saying that a lot of the configurations for a single domain protein have been already recorded in a protein database. So, so maybe like the problem itself for a subset of the problem for a subset of this data isn't that complicated. I think that's what this one's saying.
I see.
But nonetheless, extending from a single domain protein to a multi-domain protein is pretty complicated. I remember there was another paper that talks about AlphaFold's performance on those like inter-domain region, and then that might not have been good, but I don't know. Let me see.
When you say multi-domain, is it something like multimers?
I don't think it's called multimers.
Same thing? Okay.
Yeah, because multimers would be, I might have confused with the hemoglobin example. Hemoglobin might have been multiple separate chains, combined together. Yeah.
Yeah. But anyways, like there is a newer paper by AlphaFold which targets what they call multimers, like multiple proteins coming together to form a bigger protein. And one of the I guess drawbacks of the first method was that it was not good at those longer chains and bigger multimers. So they fixed that.
Okay. I didn't read AlphaFold 2, but that's impressive.
There's a, wait, so it should be in the slides too, but it's called AlphaFold multimer. I'm not sure if that's what
Oh, it just came out last year.
Yeah.
Wow, that's actually pretty impressive. I mean, if you can do multimers, you can probably do a lot of, holy smoke, you can do protein and DNA as well, I assume.
It's crazy.
Yeah.
It's putting all the biologists out of their jobs.
Yeah, like, because if you think about the applications, it's not, I mean, like our human, most of the functions are carried by protein, but there's a lot of functions that are carried by the interaction of protein and DNA. So that's how you, let's say, make more proteins, for example, given some response.
Right.
And I wonder if they talk about, let's say, immunoglobulin and antigen, because you can, with the ability to predict, let's say, protein-protein interaction, you can do a lot of stuff. Like, given this antigen, what kind of antibody would neutralize it?
Right. That was the drug research and about like future viruses. That's crazy.